Good evening to everyone. So I welcome you all on behalf of the Infectious Diseases Society of India for this webinar, IDSI Global Webinar Series. So today we are going to host the second webinar of this IDSI Global Webinar Series. So today's speaker is going to be Dr. Wong Fen. So I have to introduce Dr. Wong Fen before we start the presentation. So Dr. Wong received her PhD in 2010 and underwent her postdoctoral training between 2010 and 2012 under the supervision of Professor Masanobu Satake at Tohoku University in Japan. She established the lab with interest to study the complexity of the human immune system that is crucial to counteract pathogen invasion. Her laboratory emphasizes on the use of cellular and molecular techniques to investigate the inflammatory cytokines, innate immune receptors, and signaling pathways involved in pathogen infection by pathogens, especially Chlamydia, trachomatis, and Helicobacter pylori. And he is one of the great scientists in Southeast Asia, who is actively working on host pathogen interactions. She has received the Mon Boka Kusho Next Scholarship, Japanese Society for the Promotion of Science, JSPS Fellowship, Fulbright Visiting Scholar Fellowship, and Institute Merio Young Investigator Award. She is a mother of two lovely daughters, and she enjoys reading history books and playing badminton during her free time. So Dr. Wong is also on the editorial boards of several biomedical journals, and she is currently an associate professor at the Department of Medical Microbiology, Faculty of Medicine, the Universiti Malaya in Kuala Lumpur. She has also published enormously in host pathogen interactions, especially Chlamydia, trachomatis, and Helicobacter pylori. So with this brief introduction, let me pass on the session to Dr. Wong Fen. So thank you for making it for this meeting, Dr. Wong Fen, and please, now the session is yours. Thank you.
Much for the kind introduction and for it. So first, let me just share the slide from here.
The slides are visible, Dr. Wong Fen. Yeah.
Can you see the slide now?
Yeah, perfectly fine. Yeah, the slides are perfectly visible.
Thank you very much. Okay, so it's truly a great pleasure for me to be here today to share with you some of the insight from our ongoing works, especially on this pathogen called Chlamydia trachomatis. I'll first walk you through some of the basic biology and clinical implications of this infection. And at the second part of the talk, I will highlight some findings from our lab. And I'll particularly focus on the role of a protein called podplanin in the chlamydia-infected macrophages. Okay, let's begin with the background. Chlamydia trachomatis is a gram-negative, obligate intracellular bacterium. It cannot survive or replicate outside of the host cells, which make it particularly a tricky organism to study. The expansion of this bacteria in the lab requires a culture in human cervical epithelial cells. Usually we'll do it in HeLa cells, and followed by isolation and quantification. The picture on the right-hand side shows that chlamydia, some of them are outside of the cells and some of them are replicating inside of the cells. So in total, there are 13 different chlamydia serovars grouped according to the MOM, their MOM, the outer membrane protein sequence. So serovar A to C are associated with trachoma, which can cause blindness. Serovar D to K responsible for urogenital tract infection, whereas L1 to L3 causes lymphogranuloma venereum, so a more invasive type of STD, which is usually associated with HIV infection. Chlamydia belong to the family of Chlamydiaceae. So one other human pathogen in this family include the Chlamydia pneumonia, which causes the pneumonia or respiratory tract infection. Other species include Chlamydia abortus, which causes the abortion among the livestock in the farm. Chlamydia felis, which infects the cats. Chlamydia picorum, which causes the infection in koala bear. Chlamydia murarium is the type of chlamydia which infects the mouse. So although these species infect animals, rare zoonotic cases has also been documented in humans. For example, some farmers who work closely with the cow in the farm has reported to have chlamydia abortus, whereas most associated on pet. So on epidemiology of chlamydia, it is the most common bacteria worldwide. According to WHO estimates from year 2020, there were approximately 127 million new cases globally. Female prevalence is around 3.8%, whereas male 2.7%. And the highest burden among adolescents and young adults can be seen as between 15 to 24 years old. In Southeast Asia, in our region, so around 20 million incidents were reported for chlamydia annually, which indicates that it is a severe health concern. In Malaysia alone, the local studies has shown the high prevalence in certain population, especially among the sex workers. There are 26.5% of them were infected by chlamydia. So among those subfertile or among those who show the symptoms visiting the ONG clinics, around 22 to 51% of them are infected with chlamydia. So one of the biggest challenge of chlamydia is that it is often asymptomatic. So around 50 to 70% of infected individuals, they do not show. However, the patient, the can actually cause sepsis, mucosal discharge, bleeding. The bacteria can also ascend upwards to the uterus and to the fallopian tube, causing PID or pelvic inflammatory disease. In men, it can cause urethritis. So the complications associated with chlamydia infections are among females, so it can cause miscarriage, preterm delivery, or even ectopic pregnancy. So when chlamydia
Dr. Wong, sorry to interrupt you. The voice is actually breaking. Maybe I was wondering if you could switch off your video. I mean your personal video, so that the slide alone can be made visible. So that I think the audio-visual system would improve. Yeah, this is a suggestion. Yeah. Okay. Thank you.
Is that better? So shall I continue? So
Yeah, please. Yes, please. Yeah.
So the complications among females, it causes miscarriage or infertility or even ectopic pregnancy. And when a newborn baby is born through the vaginal canal of an infected mother, the baby can get the condition called ophthalmia neonatal or neonatal conjunctivitis when they get infected from the mother. So besides, the chlamydia also increase the risk of cervical cancer, increase the risk of getting HIV infection, and also the reactive arthritis due to prolonged inflammatory condition. So the diagnosis for chlamydia today relies on the nucleic acid amplification test, or NAAT, so which detects the chlamydia genes such as the MOM or PGP gene from which in the diagnosis. As for treatment, antibody as you remain the mainstay, and most cases they can effectively the bacteria. At present, we're still lacking against Chlamydia trachomatis, so underscore the importance of continuous investigation into the bacteria. So let me move on to the life cycle of trachomatis. So the Chlamydia trachomatis is unique as it has a biphasic developmental cycle. It has infectious or rather small alimentary body, or we call it as EB, and it has a different phase called as a reticular body, or RB. So when they are outside of the cell, they are usually in EB form. So once they enter the cells, they will turn into osmotically fragile and metabolically active RB form. So in RB form, they will continue to replicate. So and their replication occurs in a structure in the cells called as inclusion. And eventually they'll turn back into EB and being released through extrusion or cell lysis. So in the presence of stressful condition, so this enter into a persistent form, or we call it as and then they persist in the infected individual. So they can turn back into RB and continue to grow once the optimal environment is achieved. So this ability to enter into persistent state during suboptimal condition, such as during antibiotic treatment, contributes to high recurrence rate of chlamydia in the infected individuals. So the pathogenesis of chlamydia begins with the transmission through sexual contact. The bacteria, in fact, enters through vagina area. It can infect the cervix, and in fact the epithelial cells and damage the epithelial cells, and it can cause mucopurulent cervicitis, as shown here. So the bacteria can also move upward. It can track the uterus, and cause the damage in the uterine cell wall. They're going to cause endometriosis. So if the bacteria enter into the fallopian tube, they are damaged, and the scarring process can cause the fallopian tube to be blocked, So thereby prevent the egg from exiting from the ovary into the uterus. So this condition can cause tubal the human cervical vagina environment has a diverse and dynamic microbial community, usually dominated by lactobacillus. So a PhD student from our lab, Heng Jun, he has conducted a microbiome study in endocervical swab collected from around 80 female chlamydia patients and compared to the control without chlamydia infection. So he has shown that relative abundance of protective lactobacilli species decreased significantly in chlamydia positive women. And this deletion is accompanied with increase in anaerobe and potentially pathogenic bacteria such as Aerococcus and Prevotella. So reduced lactobacillus can alter the pH environment in the endocervical area, whereas the increased Aerococcus compromise the bacteria, the mucosal barrier, and Prevotella is able to provide indole and tryptophan to the chlamydia, which are nutrient required for the chlamydia replication. So this shift in microbial landscape not only create a more permissive environment for chlamydia to colonization, but it can also exacerbate the local inflammation. So one of the important feature of chlamydia is that it is a tryptophan auxotroph. It cannot synthesize the tryptophan on its own, but it requires the tryptophan for its replications. So it relies entirely on the environment or the host to provide these essential amino acids. So chlamydia infection, first it will induce an inflammatory cytokine production, mainly from neutrophils and macrophage. The cytokines such as IL-1, IL-6, and TNF-alpha. And the bar chart on the right hand side show the different chlamydia antigens is able to induce IL-1 beta from the PBNC from the patient. So besides that, adaptive immune response, including the T cells and B cells, also play essential role in fighting chlamydia infection. T cell response is predominated by T helper 1, which release the interferon gamma. Interferon gamma is important in chlamydia defense because it activates the enzyme called IDO1. So IDO is important to deplete the tryptophan store and thereby starving the bacteria, and the bacteria cannot replicate. So Chlamydia trachomatis has also evolved an equally clever countermeasure to evade immune response. So we just mentioned that the interferon gamma is able to produce IDO, which degrade the tryptophan. But in the case of chlamydia, they are able to produce tryptophan synthase, which is called as TRIP B and A, which can convert the indole into tryptophan, so they can induce the production of tryptophan to facilitate their replications. So in addition to that, there are other multiple aspects of how they evade immune system. So on B, section, can see that the chlamydia is able to reduce the phagocytosis by relocating the NADPH oxidase from the phagolysosome to the inclusion body. So, and I stop the ROS or NO production. So besides, chlamydia is also well known to inhibit the whole cell apoptosis and allowing it to persist for a longer time in the host cells. So chlamydia also block antigen presentation and delay the T cell recognition and response. So like many other successful pathogen, they can block the MSC presentation of their antigen on the surface of the infected cells. So besides, it also promotes the expression of PD-1, which is able to alter, so this is able to bind to the PD-1, the checkpoint protein, and switch off the T cell response. This similar strategy is also being utilized by cancer cells to evade the T cell response. So moving on, in murine models of chlamydia infection, we usually a species mimicking the chlamydia trachomatis in human infection. So when we infect the mouse with Chlamydia murinarum, what we found is an acute phase of inflammation which occurs within the first 20 days of the infection. So we can see a transient splenomegaly, which will peak at around 9 to 12 days, and then it will dissolve at day 20. The total number of cells in the spleen can be seen in the bar chart on the right-hand side. And besides, the total number of cells in the genital tracts are also increased transiently and continue to persist for up to 40 and 60 days of observation. Around 40 days to 80 days, we can observe the tubal damage and hydrosalpingo formation in the genital tract of the infected mouse. So when we stain the cells with the, some markers, we can see increase of macrophages in the chlamydia infected mouse in the spleen as well as in the genital tract. So using the markers, we can identify conditional macrophage and also inflammatory macrophages and dendritic cells. So both of conventional and inflammatory macrophages are increased, especially in the spleen and also in the genital tract during the day 6, day 9, and day 12. So interestingly, we know that these macrophage, especially those which are influx into the genital tract, they express a high level of a protein called as portoplanin. So this protein is usually absent or expressed in very low level in the resting macrophage, but upon infection with chlamydia, they express a very high level of portoplanin. So the bar chart on the right-hand side show the percentage of positive portoplanin positive cells. So especially the inflammatory macrophage has a high percentage of the portoplanin positive cells in the genital tract across the time of infection from day 0, day 9 up to day 60. So we're interested to further examine this expression of portoplanin on macrophage. So we have extended the study from mouse model to human sample. In human sample, the circulating blood, we can classify the monocyte into classical, intermediate, or non-classical monocytes. And we can also use some markers to distinguish M1 and M2 macrophages. So the percentage-wise, there's no difference between chlamydia negative and chlamydia positive individuals in terms of this few cell population. However, when we examine the portoplanin expression in the monocytes from this individual, you can see that the chlamydia negative individual has a low expression of portoplanin. However, in the chlamydia positive individual, they have a higher level of portoplanin. So especially in the blue color pig, which is the intermediate intermediate monocytes. So this classical monocyte has shown an increase in the portoplanin level as well. So this finding has led us to further investigate the portoplanin function in chlamydia infection. So portoplanin is a 30 to 43 kilodalton mucin type glycoprotein. It's a transmembrane protein. So it has a transmembrane domain, extracellular domain, and intracellular domain. Its extracellular domain contains a plaque domain, which is very important in the portoplanin as it can bind to the CLAC2 receptor on the platelet. And this interaction between portoplanin plaque and CLAC2 receptor can induce the platelet activation and aggregation, and is important during the inflammation and tissue injury. So in normal physiology, the portoplanin is crucial during embryogenesis. So the mouse deficient in portoplanin, they are embryonically little, and they have malfunction in the heart, lung, and also the lymphangiogenosis. In cancer, it has also shown to play a role in metastasis, especially in squamous cell carcinoma and glioblastoma. So in our context of genital chlamydia infection, we found that portoplanin expression is significantly upregulated in macrophage, especially those that are influx into the genital tract. So it is also important to note that the resting macrophage do not express portoplanin. So this has suggests that they might have an important role in immune response to chlamydia and possibly linked to some function like phagocytosis, migration, or inflammatory signaling. So to further investigate the function of portoplanin in macrophage, a student, Ying, she has generated a portoplanin knockout macrophage using the CRISPR-Cas9 vector. And we have a clone and selected the clone and established the stable clone. And using sequencing, we can see an early truncation in the knockout clone. And when we examine using the Western blot, we can see that Chlamydia trachomatis infection induces high expression of portoplanin. However, in the portoplanin knockout cells, there's no expression of portoplanin following the knockout. So using these stable cells, we examine some macrophage identity. So wild type and portoplanin knockout cells, wild type has a high protoplanin expression following chlamydia infection, but the knockout has a low protoplanin expression. But other than that, we examined a few markers like CD11B, 86, and so on. There's no significant difference among these two cells. So when we infect the cells using some fluorescent bacteria, the bacteria are able to be picked out. That means that the phagocytosis of the cells are intact. And we conducted an intracellular killing assay. We found that there's no significant difference between wild type and protoplanin knockout cells. So we continue on with the migration assay using the Boyden chamber assay. So in the chamber, we place the macrophages, either wild type or knockout cells. And at the bottom, we place the Chlamydia infected cells. So our result shows that wild type cells are able to migrate across the well, the membrane. However, in the knockout protoplanin knockout cells, the migration was significantly impaired. So next, this is the time-lapse video of wild type and knockout cells in the wound healing assay. So from this video, it's quite apparent that the wild type cells move actively to seal up the gap. However, in the knockout cells, it displays a lack of cell movement. So this suggests that the protoplanin might be important in controlling the cell migration. And in addition to migration, we also noticed that in the knockout cells, they appear in a more rounded shape, whereas in the wild type cells, the cells are bigger and they can show an elongated shape during the cell movement. Okay, when we stain the cells using F-actin in immunofluorescence staining, we can see here that in the wild type, a resting macrophage appear round, whereas upon stimulation with LPS or upon infection, they usually become bigger, and some of them will show the cytoskeletal elongation structure called as philopodia. But in the case of protoplanin knockout cells, we cannot observe such philopodia formation. What we can see is just a short arm, which might be the lamina podia, but there's no philopodia formation in the knockout cells. So we continue to investigate this phenomenon using retroviral transduction to atopically overexpress the protoplanin. So the TG means the transgenic cells, which express a high level of protoplanin compared to the wild type cells. So very interestingly, we observed that the transgenic expression of protoplanin resulted in the macrophage with explosive appearance of the philopodia. There's extensive formation of philopodia on the surface of the cells, even in the absence of any stimulation. So these highlight important roles of protoplanin in regulating the cytoskeletal dynamics during the infection. So we have done a little bit more on the using the pull down assay or IPMS. So we pull down or immunoprecipitate the protein using anti-protoplanin antibody. So in the control wild type or protoplanin transgenic or protoplanin knockout cells. And we did a gel extraction followed by the LCMS identification. So interestingly, we found that a protein called as coffilin 1, it has a high binding amount with protoplanin in the protoplanin TG transgenic cells, but not in the knockout cells. So this suggests that coffilin 1 might bind to protoplanin and cause the actin remodeling. So coffilin 1 is a molecule which is important for actin remodeling. So we predict that it might have the protoplanin might control the cell cytoskeletal dynamics by binding to these molecules and thereby enable the protrusion of the filopodia in the cells. So to summarize the talk, Chlamydia trachomatis remains a prevalent and under-addressed pathogen, especially among women and of reproductive age. So protoplanin is a protein that is upregulated in infected macrophage, but it is not expressed in the resting macrophage, which is very interesting. And our data show that it is critical for cell migration and potentially done that through interaction with coffilin. So our other data also show another molecule called filamin C, but I didn't show the data here. So as a model, we think that the chlamydia through LPS infection, it might cause the upregulation of protoplanin, and this protoplanin with coffilin and some other actin remodeling molecules and regulates the filopodia formation and enables the macrophage migration into the genital tract and initiates the inflammatory response in the genital tract. So I would like to thank all the collaborators and students involved in this work. So especially Heng Jun and Yi Ying and Yi Ting, who have done part of the work presented here. I'm so grateful to our international collaborator, Professor Bernard Arudanandam at the Tufts University, and Dr. Risin Gupta at UTSA, and Professor Yu and Professor James Chamber at UTSA. And with that, I thank you for your time and attention.
Thank you. Thank you, Professor Wong Fen. Thank you so much. That was a very that is a wonderful presentation. So we really enjoyed your presentation. That
Thank you
was
so much.
truly, humongous data that we have generated. And thanks for enlightening the audience with your excellent presentation. So thank you so much. So we have some questions that have been raised by the audience. Maybe I can read out. Of course, we have some 4 or 5 questions. Maybe I can ask some of those questions. So one, Mr. Atesham Hanifi is asking whether there is any CRISPR-Cas13 based therapeutics against Chlamydia trachomatis. CRISPR-Cas13.
Okay, thank you very much for the question. For the CRISPR-Cas13, I've encountered a paper published not long ago, which has use the CRISPR-Cas13A in the diagnosis of chlamydia.
Okay,
Yes,
so that's a modality that is currently being tried against Chlamydia trachomatis.
yes.
Okay.
So it's an interesting new tools that is used for diagnosis.
Okay. And the second question is from Dr. Deepak Jayakumar from the State Public Health Laboratory in Chennai. So he is asking, can the genes encoding for TRPBA be knocked out in Chlamydia trachomatis? If yes, will that strategy attenuate virulence in the organism? And has any work been done in this area? So that is what is his question.
Wait, sorry, can you repeat again? The genes encoding for?
He's asking if the genes encoding for TRPBA be knocked out in trachomatis, Chlamydia trachomatis. If so, whether the strategy will attenuate the virulence in this organism?
Yeah, that is a really interesting question. That in the Chlamydia trachomatis, making a mutant is rather challenging. So I haven't seen a report on mutating the TRPAB gene yet, but some of the report has utilized the T3SS has created some mutation in the T3SS subunits. That's been reported by Mary Webber's laboratory in USA. So, but in the case of tryptophan synthase, maybe there's a great potential to further study the genes in the future.
Okay. And we also understand that there is no vaccine, active vaccine available against Chlamydia trachomatis.
Right.
Yeah. And the next question is from Dr. Selvaraj Jagannathan from Pasteur Institute. He is asking, does chlamydia has any role in combination with human papillomavirus in increasing the risk of development of cervical cancer?
Yes, the reports has shown that HPV and chlamydia co-infection does increase the risk for cervical cancer.
Okay, but does Chlamydia trachomatis has any direct role in causing cervical cancer, like what we have for human papillomavirus?
So I'm afraid not, but it is through the chronic inflammation that eventually facilitate the
development of cervical carcinoma.
Yes.
Okay, and so another person from audience is asking, it is mentioned that the LPS activates chlamydia infection and our gut is full of LPS of gram-negative microbiota. So can you please tell me how chlamydia infection is getting prevented in our gut?
The chlamydia infection is
prevented in our gut. I think
okay.
I don't know in what context.
Yeah. So it's in the vagina. Is it in the vagina?
Yeah. The context is genitourinary chlamydia infection, but
yes, right.
He's asking whether chlamydia infection is getting prevented by microbiota in the gut.
Yes, microbiota in the vagina area. Yes, because the microbiota diversity, especially the presence of the lactobacillus, it helped to protect the area from being colonized by any other pathogens, including the chlamydia.
So sexual promiscuity will deplete lactobacillus, and then it paves way for acquisition of Chlamydia trachomatis infection. Am I right?
Yes, that is what
Yeah.
is shown in the microbiota data, in which the Chlamydia trachomatis positive individual, they have less lactobacillus, and they have increased amount of other pathogens.
Okay. So Dr. Saktivel Govindaraj from US, from Emory University, US, is asking, is there a window period during the menstrual cycle when Chlamydia trachomatis is more prevalent? If so, which phase of menstrual cycle makes individual more vulnerable to infection, acquisition of infection?
This is more to the human menstrual cycle, so I'm afraid I can't answer this question. I'm so sorry.
Yeah, I know. And next one is from Dr. Satish Naik, who is from the Central University of Tamil Nadu. How does Chlamydia trachomatis interact with host cell surface receptors such as PDGF receptor and EGF receptor to facilitate entry and causing infection during the early stages? Any particular mechanisms or pathway involved? So this is what is his question. He is asking more about the interaction between the host cell and the Chlamydia trachomatis. What kind of receptors does the reticulate? I mean the reticulate bodies, right? Elementary bodies
Elementary
utilized
body.
to enter into
Yeah.
elementary bodies to enter into the cell. That is what is his question. Receptor, any specific receptors like the PDGF receptor or the EGFR, which can facilitate its entry into the host cell. He wants to
Okay,
know the
thank you
receptor.
very much. Okay, so first of all, maybe let me answer the question just now on the human menstrual cycle. So on human menstrual cycle, right? So in the mouse model that we use, we often inject the mouse using the depot progesterone. So we will inject the mouse with progesterone for a week before we start the infection. So the high level of progesterone assist in promotes the chlamydia infection in a better way. So, okay, so on the second questions on chlamydia adherence and chlamydia receptors, which facilitate its adherence, there are certain molecules that are being identified, including like afferent receptors, like which are important for the chlamydia adherence and entry into the cells.
Okay, so the other question from the audience is, what are all the wound healing assays that have been done for this?
So we conducted the wound healing assay because we are trying to examine the macrophage, the effect of the autoplanin knockout on the macrophage cells. So we suspect that this autoplanin control the cell migration during the chlamydia infection. So we compare between the wild type and the knockout cells. And what we can see is that the cells without the autoplanin, they fail to migrate to the empty spaces compared to the wild type cells. So this suggests that the autoplanin control the cell migration towards the infection or injury site.
So another, again, one more question from Mr. Satish Naik from Central University. Is asking, is there any role of the type 3 secretion system in Chlamydia trachomatis pathogenesis? Does it deliver effector proteins into host cells?
Yes, so type 3 secretory system is important for Chlamydia pathogenesis. So it has been substantially studied by some other groups, and which we are not studying the type 3 secretory system in our lecture.
Okay. So the other question from Dr. Selvaraj Jagannathan is, is appreciating the presentation is lucid, and is asking if the outer membrane protein of this pathogen, will it become a conserved, because it's a conserved protein, will it become a suitable vaccine candidate?
So in some other for some other species like the Chlamydia abortus, Chlamydia picorum, which affects the koala bear, Chlamydia abortus, which affects the livestock in cows or sheep. So in this species, the mom has been shown to be a successful vaccine candidate, which can help to protect from abortion, from the Chlamydia abortus or Chlamydia picorum infection. So it has been shown successful in animal model. However, in human, unfortunately, it is not. It has not been. It has not been a good candidate so far. Probably the reason why, first of all, we have many different serovars, up to 13 different serovars with different mom sequence. So in that case, although it has been a successful vaccine candidate that is being used, and it is so far the best recognized candidates in vaccine as well, but it's not been successfully translate into a vaccine at the moment.
Okay. So this is the question from Dr. Marie Larsen from Linköping University, Sweden. She's asking, do a resolved chlamydia's infection protect against reinfection?
So not in all the cases, because the chlamydia is well known to enter into persistent phase. So once it enters into persistent phase, it can stay in the patient for a long period. So even in the presence of antibiotics, so in the presence of antibiotics, the bacteria will be under some stress environment, and then they will choose to enter into a their body. So they do not wrap it, but they just stay in the epithelial cells. So once the condition allows, there will cause infection in the patients. So the antibiotics may resolve the case, but in some cases it may not be able to fully resolve or prevent future recurrence.
Okay. So the other question from the audience is there any cure for this Chlamydia trachomatis infection?
Is there any
Cure.
cure?
Yeah.
Cure. So the best cure will be the antibiotics. So antibiotics can cure, can kill the bacteria, but the main problem for chlamydia is that usually they will be asymptomatic. When they are asymptomatic, it encourages the transmission from person to person unintentionally. So especially among the young adults, adolescents, they are not aware or they do not practice the sex protection. So they might acquire the chlamydia unknowingly. So the best way to prevent chlamydia would be a vaccine compared to antibiotics, because usually when the symptoms show, it is too late because the damage at the epithelial layers are already done, the damage to the fallopian tube already done, and that affects the female fertility.
Okay, so the other question is there any specific confirmation test for trachomatis?
Confirmation test, it will be the nucleic acid. So nucleic acid amplification test is the gold standard for the diagnosis of Chlamydia trachomatis. We do use the PCR amplification. So usually we need 2 rounds of PCR amplification or nested PCR because usually the chlamydia is quite hard to be detected by one single round of PCR. So we isolated the DNA from endocervical swab or vaginal swab or urine. So isolate DNA, and then we run 2 rounds of PCR using the MOM primers or PGP, some plasmid primers, and we run a gel electrophoresis to check the presence of the bacteria. So in addition to this, IgM or IgG ELISA can be done for chlamydia diagnosis.
Yeah. So this is the final question, and this is from Professor Ramendra Pandey from SRM University, Haryana. He is asking, is there any specific correlation between pro-inflammatory cytokines and the chlamydial infection? Do neutrophils play any role in the protection against Chlamydia trachomatis infection?
Okay, neutrophil play a very important role in chlamydia infection because at the acute phase of infection, a large amount of neutrophil will enter the genital tract. So you can detect a huge amount of neutrophils at the genital tract during the acute infection phase. And then there has been a report which shows that chlamydia is able to block the neutrophil net formation, which is neutrophil extracellular tract formation. So some study has been done on chlamydia and neutrophils and shows that chlamydia is able to block neutrophils, and thereby they are able to cause further infection in the patient.
Dr. Wonfen,
Yes.
I have one question.
Sure.
So in your study, you have said that IDO can inhibit the IDO that is generated from breakdown of tryptophan can inhibit Chlamydia trachomatis. Is that
so? IDO can inhibit the tryptophan because the tryptophan is required for chlamydia to replicate. It is
to replicate? So
yes.
it's a chlamydia. I mean, it's a chlamydial. I mean, it's an auxotroph. Chlamydia is a tryptophan auxotroph. Yeah.
Yes, exactly. Yeah.
So if that is the case, then you also see that IDO can also inhibit T cell functions, right?
Mm-hmm. Yeah. Mm-hmm.
So that way you think that chlamydia is chlamydia. I mean, chlamydia's persistence can be inhibited, as well as T cell function also parallelly gets inhibited. Am I right?
Yeah.
Because
right.
Tryptophan is an amino acid that is required also for T cell metabolism. It's an
Yes,
important amino acid.
exactly.
Yeah.
Yes, you're right.
And then about the macrophages, you said conventional macrophages. Is that is it something like the M1 macrophage or that you were mentioning?
Oh yeah. So inflammatory macrophage refers to M1 macrophage, but it's
M1 macrophage. Okay. Yeah.
equivalent to humans M1 macrophage.
Oh, okay. That was fantastic. Yeah. So I think there aren't any more questions from the audience. Yeah, if there aren't any further questions, then maybe we can conclude this session. So I must thank on behalf of the Infectious Diseases Society of India, Associate Professor Dr. Wong Fen, who
Thank you
also
very
happens
for the
also happens
opportunity to share the work, and thank you for all the questions raised for discussion.
that was very much enlightening, and it was nice. Maybe we will also invite Dr. Wong Fen for the next upcoming conference that will be held in Chennai in 2027. The ISRID 2027 is planned to be hosted in Chennai. So the IDSI, the Infectious Diseases Society of India will be happy to honor you by inviting you, for the conference.
I'll be glad
So
to.
it was, yeah. Thank you, Dr. Wong Fen, and thank you so much for honoring us with this wonderful presentation and for patiently answering all the queries raised by the audience. So I hope that the audience might have enjoyed the lecture, and also they might have enjoyed the interaction with the speaker. So with this, I would like to thank the audience as well as the speaker of this for this international, Infectious Diseases Society of India's second global seminar, global webinar series. So I would like to thank all of you once again, and wish you all the best. Thank you so much. Thank you, Dr. Wong. Thank you.
Thank you so much.
We will see the next webinar series that will be hosted on May, I think May 4th. May 4th. Yeah, May 4th by Dr. Muhammad Rizwan, so who will be from the Ministry of Health, Government of Malaysia. Yeah. Thank you so much, and have a wonderful evening. Thank you. Bye-bye.